Okay, so hello everyone. Thank you for joining the Atto Fridays today. We're going to have the last session of our season of 2022. And this is going to be a hot topic session with early career researchers. And it's a pleasure to have the three of them here. We're going to hear from Xinyao, Simon Brennecke, and Antonia Freibert. It's going to be focused mainly on machine learning, but we are also going to have a nice X-ray session. So thank you all for joining and supporting the early career researchers in our field. We are looking forward to the talks. And the first one who is going to speak is Simon Brennecke. He is a PhD student in the group of Manfred Lein in Hannover, Germany. And he's going to talk about two publications in which he looks at photoelectron holography, and he's going to do the quantitative modeling of interference in this situation, and he's going to argue about the subtleties that one needs to take into consideration. And also, he's going to show how you can deal with inversion problem, mapping from final to initial conditions. Actually, it's interesting because we started inverting and now we're trying something forward, and they started forward and now they are inverting. But this is not a trivial matter, and we're looking forward to hear about how one can use clustering to solve the inversion problem and to make it versatile. Okay, so if you're ready to go, we are. Feel free to start. Afterwards, we're going to hear from Antonia Freibert and Xinyao. Looking forward to the talks as well.
Can you see the screen well?
Everything is fine, yes.
Then thank you for the kind introduction and for the invitation. So I'd like to talk about the Coulomb lens and strong field ionization. And I think probably most of us, when they think about a lens, they think about classical optics of light waves. For example, a Gaussian light beam that is focused by a lens to a point, which means a 2D manifold of optical rays is focused to a point. And then, of course, when we look at the intensity across the focus, the intensity changes and peaks at the focus. And we all know that at the same time, also the phase of the beam changes, and this phase change is known as Guy's phase anomaly. And if you now zoom out and look from far away, much larger than the Rayleigh length, then this phase change looks like a phase jump of pi. And of course, we do not only know light waves, but also matter waves or acoustic waves. And hence, we would expect that in these types of waves, there are similar phenomena. And in this talk, I want to talk about electron waves produced by strong field ionization. So we first consider strong field ionization in linearly polarized fields, which means we have atoms in short, strong, typically femtosecond laser pulses with wavelength around 800 nanometers or even longer, and field strengths which are high enough that the electric field strength of the laser field is in the same order of magnitude as the electric field that binds the electron to the core. And in this special strong field situation, we know that the ionization process can be described by the famous two-step model, which means that electrons are first liberated by either tunneling or multiphoton processes. They appear then in the continuum at certain initial times with initial velocities. And then they can be often well described by classical particles, which are driven by the laser's electric field, but typically also influenced by the ionic Coulomb field. And an example of a trajectory is, for example, shown here. So the electron appears at the tunnel exit. For this special time of release, it is first driven out by the electric field, then the electric field switches its sign, so it's driven backward, it scatters, so it's strongly influenced by the ionic core, and hence it is deflected. Of course, such a single trajectory is not is not it is not unique. There are many trajectories. For example, you cannot only consider this initial velocity, but you can rotate the initial velocity around the polarization axis here in linearly polarized fields. And then you get the surface here of trajectories, which means that along each angle you have one trajectory, so you have a 1D manifold of trajectories. And importantly, all these trajectories are focused here when they pass the polarization axis in the same point. So we have also some focusing, some Coulomb focusing of the electron trajectories here in this strong field ionization process. And hence somehow this Coulomb field acts like a lens. And in agreement or similar to optics, we of course expect an enhancement in the ionization probability, which was already predicted in the 90s. There were also enhancements seen in the photoelectron momentum distribution. But what we also expect is that when such an electron wave passes here a focal point, that it phase jumps or it phase changes. And this is this well-known Guis phase anomaly. Importantly, such focusing in this sense here only appears in 3D, because here we need this 1D manifold of trajectories. If you consider the 2D situation, we only have two trajectories which cross, and this is not the focal point. So we expect different phases after focusing between a 2D situation, or this typically often in theory considered 2D situation compared to the real 3D situation. Of course, we cannot directly measure a phase of an outgoing electron wave packet, but we can interfere two electron wave packets and consider the interference pattern. And a quite famous one is here the photoelectron holography, more precisely the spider type interference, which was already discovered by Huysmans 2011. And here we have these interference fringes, which are roughly parallel to the polarization axis. And they are actually caused by the interference of a reference wave packet of direct electrons, here shown as a blue trajectory. And this blue trajectory is only weakly influenced by the core. It does not pass a focal point. And it interferes with a signal wave packet of rescheduled electrons. And these are exactly this focused electrons which are introduced before. And hence, of course, in the interference pattern, their phase difference determines the exact fringe positions. So let us look on this situation from a theory perspective. To this end, we solve numerically the time-dependent Schrödinger equation for helium 800 nanometer. And for simplicity, we use a short 2-cycle laser pulse. However, the things I will present in the following are also correct for longer laser pulses, but then, of course, ATI rings and so on make the analysis more complicated. And then we did one calculation in 2D and a second calculation in 3D, where we consider here a slice through the 3D photoelectron momentum distribution. And I think two things are quite prominent. The emission strength in 2D and 3D is different. And in addition, if you look here at the interference fringes, for example, this holographic pattern roughly parallel to the x-axis, then you see that in 2D, the central fringe is much smaller than in 3D. So the fringe positions are different in 2D than in 3D. And this is already a qualitative sign of this Coulomb focusing and the associated GUI phase in 3D. However, we want to model it quantitatively. And I think in strong field physics, there is already a zoo of models. In particular, successful to model holography are models based on this path integral. So these models in this region here in this schematic illustration. And hence also our model follows roughly the ideas of this previous models. However, what is missing in the models? To look at it, what we do is we use a semi-classical approximation to Feynman's path integral and follow the idea of the two-step model. So in the first release step, which we treat with strong field approximation, the electron is released, and there the initial conditions for the later motion are defined. So we have some release times t0, some initial velocities, and some tunnel exit position, and we can parameterize them in the spirit of SFA by means of an auxiliary momentum p prime. Afterwards, we treat the motion classically. So we propagate classical trajectories, which are governed by the laser's electric field as well as the Coulomb attraction. And then the photoelectron momentum distribution can be calculated here as this sum over the different trajectories. And I would say two terms are quite familiar. One is this action term, where the action is actually collected along the classical trajectory. And the second one here is an ionization matrix element. And the modulus squared of this ionization matrix element is somehow the ionization probability. But then we have this middle term here. And this middle term I called here a prefactor, and it has two important ingredients. One is the Jacobian, or more precisely, the modulus of the Jacobian, and this determines the weighting of the trajectories or influences this weighting. And then the second one, even more important for our observations, is the Maslov index nu, and this represents phase jump as focal points. I will consider these two ingredients in the following more precisely. So when we look here at the sum, we have to sum over all trajectories with a given final momentum p, which means we have to determine all initial conditions, here represented by this p prime, that are mapped by the classical dynamics to the given final momentum p. However, this is typically a quite cumbersome task, because what we can do easily is to solve the initial value problem, which means we give some initial conditions here, propagate our classical trajectories, and get a final momentum. This is done easily. But what we need to do is, we want to look at one final momentum, and we need to find all initial conditions that are mapped to this final momentum. And in general, we do not even know how many initial conditions that are, and we do not know at all where the initial conditions are placed here in this plane. So we thought about it, and we came up with one possible solution. So one solution would be, of course, simply use Newton methods or something like that to solve this inversion problem. But this has some difficulties. So we invented a different method. And to this end, what we do is we sample initial conditions, shown here. Then we calculate the trajectories. We get some final momenta, shown here. And we do it with many points. So we somehow scan this function d, which map the initial conditions to the final momenta. And we do it again and again and get many, many points. And then we want to consider one final momentum in the space of final momenta and look at a small bin around it. And if you only consider those trajectories which end up in the small bin and consider their initial momenta p prime, then you see that here some small agglomerations are formed. For example, here four small agglomerations. And it is important that these are not 4 points, but these are 4 agglomerations of many, many, many points, as you can see here in the magnification. And now the important thing is you have to identify those different agglomerations. And now here the machine learnings come into play. So what we use is a clustering algorithm. This can be classified as unsupervised machine learning. And this clustering algorithm takes this initial points p prime here, all these points. And what it does, it identifies the clusters. For example, here afterwards, after the clustering algorithm has identified the clusters, I can color mark them, for example, here in red, blue, green, and orange. And we in particular use such a density-based spatial clustering with applications of noise algorithm, which is, for example, implemented already in Python and works quite well. So what do these clusters physically mean? These clusters physically correspond to different types of trajectories. For example, all these points here in the blue cluster have nearly the same shape, and it is a direct trajectory. So the red cluster is associated with such a rescattering trajectory we discussed before. So when we have identified the different clusters, we have identified the different trajectories which interfere with each other and form the photoelectron momentum distribution. So we can use out of each cluster one representative trajectory, and then we have solved the inversion problem. So now we know which trajectories interfere with each other, but in order to calculate our photoelectron momentum, we know which trajectories interfere, and we cannot only do it for linearly polarized field, but this clustering method is relatively versatile because you don't need to add in some physical intuition. You simply first shoot your trajectories, then you apply the clustering method, and then you get out the clusters, and hence you have a solution of the inversion problem. And here, for example, we used a bicircular field, and also here we can identify the clusters. Here we have more clusters, 10, and it works quite well. So now we have identified our initial conditions and the trajectories, but what we need to calculate actually the photoelectron momentum distribution is the Jacobian and the Maslov index. And probably we are all aware of having a trajectory and associating a semi-classical phase to the trajectory. And for the Jacobian and the Maslov index, we can now associate a small volume element in momentum space to the trajectory. So we have here a trajectory in momentum space shown in red because we want to evaluate in the end photoelectron momentum distribution. We choose here a description momentum space, and we associate a small volume element to the trajectory, and then we look how this volume element evolves in time under the classical evolution. And you see that, for example, for this trajectory, it changes. And this change of the volume element in the end, well, the associated oriented volume is then this Jacobian J here. And now you see here for the blue situation, you have a very special point because there the volume vanishes. So J is 0. And this vanishing volume indicates a focal point here in momentum space. And for such a focal point, you find a direction in the space of initial conditions or a zero mode, such that when changing the initial conditions in this direction, in this example, for example, along the x-axis, your momentum at this focal point, capital T, does not change. So it's here the same. And hence, because of this, it's called a focal point. But now the important thing is that the Maslov index can only change at this focal point. There it jumps. And hence, this Maslov index exactly represents this Guis phase anomaly, which we know from optics. And to calculate the jumps of the focal points, you can, for example, consider this paper here. So this was quite abstract. So we can, for example, consider the special case of linearly polarized field. In linearly polarized fields, we know that we have this nice rotational symmetry around the polarization axis. So here the x-axis. So if we want to calculate the photoelectron momentum distribution in one slice through the PMD, we only need to consider classical trajectories in this plane. For example, here the XZ plane. Which means in 2D and in 3D, whether we consider the 2D or the full 3D dynamics, we have the same trajectories. However, the associated Jacobian and Maslov indices are different in the 2D and in the 3D situation. And this exactly resembles this modulation of the amplitude due to Coulomb focusing and the modulation of the phase due to Coulomb focusing. For example, here for the Maslov index, in addition to the 2D Maslov index, in 3D, we have to count the number of zero crossings of this lateral momentum component along the trajectory. And this means that in general, a slice through the PMD in 3D, not even for linearly polarized light, is equal to the PMD in 2D. And hence, because our model does include this important properties of the prefactor, our model can reproduce this difference between the 2D and the 3D TDSE, which we saw before. So we see here a different emission strength in 2D compared to 3D. And what we also see is when we look here at the fringes, that the fringes look different. And we will do it here now in detail. So as said before, the holographic pattern is formed out of a reference wave packet and a signal wave packet. And also here our momentum space analysis showed that this direct reference wave packet has Maslov index 0. However, the scattered wave packet, there the calculation indeed showed that in 3D it has Maslov index 1. In 2D, because there's Coulomb focusing absent, it has still Maslov index 0. And this momentum space description in the classical model is indeed in agreement with the simple position space intuitive interpretation I gave in the beginning. Hence, if we look here at slices through the PMD, in 2D, one can nicely describe the TDSE when when neglecting Maslov space in the semi-classical calculations. However, in 3D, here the fringe positions are systematically off, and only including Maslov's index, or only when Maslov's index and hence Gui's phase anomaly is included, one can correctly predict the interference fringe positions. So till now, we only considered linearly polarized fields. And hence probably Coulomb focusing looks like something quite special, but it is not. So Coulomb focusing is a quite general property in strong field physics. As long as one has a strong interplay between the ionic core and the driving electromagnetic field. And to illustrate that, we can consider here the example of a bicircular counter-rotating field with a relative intensity ratio of 3, such that the field looks like this here in this schematic illustration. And for such types of field, rescattering is quite important. And it has already pointed out theoretically and shown experimentally that in these types of field also interferences appear in the photoelectron momentum distribution. And here I show again a PMD from numerical solution of the TDSE in 2D and in 3D. And you can again see that there are differences in the 2D compared to the 3D PMD. For example, if you consider here the interference fringes, they look here relatively different in the 3D situation compared to the 2D situation. If we now use our model, we can indeed reproduce this difference quite nicely. And this is because, again, here in 3D, we have Coulomb focusing, and in 2D, we don't have Coulomb focusing. To look at this in a little more detail, we can consider here this interference fringes, which are roughly parallel to the negative px axis. So we look at these fringes here. And the analysis then showed that these fringes are caused by interference of a direct non-scattered trajectory, which is shown here in blue. And the actual analysis then also revealed that the associated Maslov index is 0. But we have also here a scattered trajectory with, in 3D, a Maslov index of +1. And again, the scattered trajectory passes a focal point. And to see that, we can here again use this position space description. And instead of choosing the initial conditions for the red trajectory, which is in the polarization plane, we can start other trajectories with a slight initial velocity in z direction. And then first these trajectories here diverge, but then they are focused to one point, they pass this focal point, and afterwards they diverge again. But after passing the focal point, they again have collected a Gui phase. Importantly, for this more complex, non-trivial situation, we don't have a simple formula for the 3D and Jacobian and 3D Maslov index, but here we really have to evaluate the complicated formulas. But when we then consider the interference pattern for these two trajectories, it is shown here, and we have indicated here the lines of minima, either when we include Maslov, the difference in the Maslov index, or we neglect it. So included is the black dashed line, if we neglected the gray dashed line. And when we now compare it to the numerical solution of the TDSE in 3D, we see that the black dashed line again matches much better the line of minima, for example, here, than the gray dashed line. So we also see for this non-trivial geometry the influence of Gui's phase anomaly on photoelectron momentum distribution. And to go one step further in our most previous work, I would say, we do not only consider strong field ionization, but we considered the interplay of electron wave packets driven by terahertz pulses and the Coulomb field. And even in such strange looking situation, one can find rescattering phenomena which are quite similar to holography or Coulomb focusing, but under really scaled conditions. And yeah, because we used here a two pulse scheme consisting of a short IR pulse and then a terahertz pulse, by controlling their delay, we could really control the dynamics and also the scattering dynamics to see, for example, for the given delay here, quite nicely also some focusing effects, like in usual strong field ionization, but as I said, under really scaled conditions. And with this, I'd like to conclude. So what we saw is there is Coulomb focusing in electron waves and strong field approximation. I mean, this is not really news. And also already earlier, it was known that the amplitude, so the emission strength of the electrons is modulated by Coulomb focusing. But what we found is that also the phase jumps at this focal point, and hence the interference patterns are modulated by Coulomb focusing. And in order to model it, we developed, or we further developed other semi-classical models and included this prefactor consisting of the Jacobian and the Maslov index. And here the correct weighting with 1 over the square root of the Jacobian is important for the correct signal strength, and the inclusion of the Maslov index, and hence the inclusion of Gui's phase anomaly, is important to reproduce the correct fringe positions in 3D due to the Coulomb focusing. And the implementation of our model was only possible because we were able to solve the inversion problem, which means we were able to determine all initial conditions that were mapped by the classical dynamics to a given final momentum. And to this end, here we used machine learning and we used clustering algorithms. So thank you for the attention.
Thank you so much for your talk. Are there questions, comments? Please put them in the Q&A tab. While we are waiting for the public to ask stuff, there is something I would like to ask you. My postdoc, Brett Augstein, unfortunately cannot be here today, but he sent us a few questions, and I cannot put in the Q&A, so because I'm the chair, so I'll put them in the chat. And he was interested in knowing how you use exactly clustering to solve the inversion problem, and why this preference for the DB-scan rather than other clustering algorithms. I mean, you could have used other ones, and he asked why these specifically.
So we tried, I would say, 3 or 5, which were quite advertised in this Python community. So I think there, if you look at the sci-fi package in Python, and the scikit-learn package in Python, then there are many different clustering algorithms implemented, but you want to have a clustering algorithm which is relatively stable, for example, if you have a cluster which only consists of a few points, and hence a clustering algorithm with noise was, or this DB scan algorithm, which includes here some, or where noise can be treated, was quite good, because then single points which were somewhere in space did not disturb the search of clusters. So usually you have, when you shoot your trajectories, you have some really strange trajectories, which, for example, circle around the core a few times. And this could disturb then the search of clusters, or we saw that it will disturb the search of clusters. So by using such a application with noise, we could avoid the influence. So this is why we have chosen something with noise, and then this density-based spatial clustering, I would say, because it worked. So we tried a few, and this one worked for us best.
Mm-hmm. And the key issue for you was noise, because it was a problem that was very noisy. And
it's not, I mean, noise is, it's not noise what we have. I mean, these are additional physical trajectories, but
okay.
I would say that they are single trajectories. So as you can see here, a cluster is formed probably out of 5 points or at least 10 points or something. Mm-hmm. But you can have some additional trajectories which also lead to the given final momentum, but they are probably quite strange. And this is important if you especially go to small initial momenta. Then the trajectories which start with small initial momenta, for example, can circle around the core a few times. Mm-hmm. I mean, they do not have really a huge weight in the end in the final electron momentum distribution, but they will disturb your search of clusters. And in order to avoid such, I would probably call them chaotic trajectories, we looked for a clustering algorithm which exactly can avoid single points, and the clustering algorithm which can avoid single points is this DB scan algorithm, because there it treats the single points, even though it's not noise in the usual sense, like noise.
Okay. Yeah, they are there, and they are not a huge amount, but they do disturb. I see your point. And I think this comes then to his next question. What trajectory parameters, like initial and final longitudinal perpendicular momentum and times, do you fit to the model, and why? Because I understand you do like a sweeping at the beginning, you launch a lot of parameters, then you propagate, and then from the bin, you propagate back and solve using the clustering. Is this correct?
Yes, more or less. So yeah, we in the beginning sample many different initial conditions, and in principle you can use whatever you want. So it gets more efficient or it gets less efficient. But as long as you sample all initial conditions, when you take in enough points, it should work. But of course, the number of points you need to solve or to sample them properly is different. So what worked for us pretty well is, for example, sampling according to the ADK rate. This at least gives a good estimate. If you want to make it a little better, I would suggest not sampling according to ADK rate, but adding to the ADK rate for example, Gaussian distribution with small lateral momenta, because the small lateral momenta in the end lead to scattered trajectories. So for large angle scattering, you need to start your trajectories with small lateral momenta. And in order, for example, to get a good resolution in the high energy plateau region, as you could see here, we could also nicely simulate this high energy plateau region. There you need large angle scattered trajectories. So in order to make this clustering method quite efficient, we started some additional trajectories with small lateral momenta. But in the end, how you sample these initial conditions should not influence your final result. So it should be independent. It can only, or what depends on this initial choice is how much time you need and how much computational resources you will need. Because the number of points you need to sample depends on the choice of the initial.
Yeah, this comes to the next question, which is, what was the improvement that you obtained by using clustering? Because I know that with direct methods, people need 10 to the 8, 10 to the 9 trajectories to get converged results. So now, how much can you reduce the number of trajectories by using clustering?
Actually, I did not try to scale it to the minimal amount of trajectories, to be honest. I think these pictures I showed here were calculated with 10 to the 8 trajectories. Mm-hmm. I would say the biggest advantage of the clustering algorithm is that it is versatile. So, for example, I would say to find the correct initial conditions for this example of the bicircular field would be quite difficult. And with clustering, we did not need to change anything. So we simply inserted the new shape of the electric field in the Newton equation, and then we started again. And in the end, a new picture came out with a new momentum distribution without changing much. And this is, I would say, the biggest advantage, that you don't have to plug in physical intuition, or you can at first avoid it. So of course, as I said before, if you have physical intuition, for example, which initial conditions are important for rescattering and so on, you can make it more efficient, but you don't need it.
I see. Yeah, you have quite a few acoustics as well. I can see also in the bicircular field. So how do you deal with bifurcations and also with these acoustics? My question is not from Brad.
Yeah, this is probably the most difficult point. I mean, acoustics are there in semi-classical approximation. So it's not related to the clustering algorithm. It's related to the approximation of the path integral. And in principle, you cannot do nothing against it. So they will be there. Okay, you can go beyond this usual approximation and then use a uniform approximation and so on. But this is, I would say, quite difficult for most problems. Hence, we did nothing basically with it. They were there, and this was okay. Of course, you get a problem if you have an initial condition which leads directly, or which is really close to the caustic, because then your spectral weight blows up in the semi-classical simulation. But yeah, then you get a bright point simply. So in order to avoid it, you could probably not use the Jacobian here.
Yeah, but then you lose the stability factor, and you lose the proper weighting, because our method has a Jacobian. We don't have the Maslov indices. We put them by hand, okay, in the linearly polarized field in some cases. But if you remove this, you will get the wrong weighting.
No, I don't mean remove it, but if you don't want to have a hugely blown up spectrum, you could, for example, not use J, but you could use J plus a small epsilon. Mm-hmm. And then you have at least cured it a little. But this is, of course, not treated properly.
Okay, so I think we should move on. So thank you so much. There will be opportunities to ask questions further after we are done with the section, but we should move. So our next speaker, and I'm glad she managed to join today, is Antonia Freibert. And she is going to talk. She's actually a former UCL student. She did the master's at UCL, and now she's a PhD student supervised by Niels Huse from Hamburg, Germany, and Oriol Vendrell from Heidelberg. And the topic is going to be quite different, but also quite interesting. She's going to talk about non-adiabatic dynamics of excited molecular systems, and is going to describe X-ray spectroscopy. And I agree, it fits quite well. So the topic is going to be simulating time-resolved X-ray absorption spectroscopy of pyrazine at nitrogen K edge with a full time domain approach. So it has everything to do with the Atto Fridays. We agree with her, and we are happy to have her here giving her talk in the hot topics today. So the floor is yours. Please feel free to start. We're all ears.
Okay. So, can you see my screen?
Yes, it's nice.
Okay.
Perfect.
Okay, great. Yeah, many thanks for the kind introduction. And yeah, first of all, thanks to all organizers for giving me the opportunity to present my work today. So yeah, as already mentioned, I'm going to talk about simulating time-resolved X-ray absorption spectroscopy of pyrazine at the nitrogen K edge with a full time domain approach. Yeah, during the past 2 decades, experimental developments of ultra-fast and intense X-ray sources made it possible to extend pump probe techniques into the X-ray domain. And this allows tracking photo-induced molecular processes by core-level probing with femtosecond time resolution, or even in the attosecond time regime. However, due to the complex nature of the underlying processes, we usually rely on computer simulations to support experimental measurements and to extract the right information from the data. And yeah, for an accurate description, we need to solve the time-dependent Schrödinger equation. But as we all know, for almost all chemical systems, it is impossible to find an analytical solution. Hence, several assumptions and approximations must be made to implement a workable method. And in this talk, I'm going to discuss 2 widely used approximations when simulating X-ray spectroscopy, namely the short time or Lorentzian limit approximation, which states that the nuclear dynamics after X-ray probing is insignificant. And further, we discuss how an explicit description of the external electric field might influence the outcoming spectra. Yeah, let's start with a little bit of theory to understand where it comes from. So when we are interested in simulating equilibrium ground state X-ray absorption spectroscopy, we can apply Fermi's golden rule to calculate the static absorption spectra. So in a bit more detail, Fermi's golden rule states that the transition probability from an energy eigenstate, here denoted by psi naught, to a final excited state psi j, is given by the overlap of the corresponding wave functions. So the peak intensity for a specific transition is just defined by this matrix overlap element. And since for example, the lifetime of the core hole states are very short, we also get a spectral broadening controlled by this dephasing factor gamma. Alternatively to this energy domain approach, we can also use wave packet dynamics for simulating absorption spectrum. So yeah, in the completely equivalent time domain approach, what we are doing is we put our initial wave function onto an excited state where there's no eigenfunction anymore, but a wave packet and let it evolve. And the absorption spectrum is then given by the Fourier transformation of the autocorrelation function of this wave packet. However, the situation becomes more challenging when starting from a transition state, and the theory behind must be adjusted. But yeah, as my title suggests, this is exactly what we are aiming to do. So we want to simulate time-resolved X-ray experiments to get a better understanding of the photo-induced molecular processes. That means the situation we now want to describe is that we have an optical pump pulse that brings our system into an excited state and triggers the photochemical dynamics we are interested in. And then we probe that photochemistry by sending a second X-ray pulse at different time delays, which allows us to observe the photo-induced dynamics from the perspective of a specific atom in the molecule. Yeah, in contrast to before, where we started from the equilibrium ground state, we now have this evolving wave packet triggered by the first pump pulse, and we want to calculate the transient X-ray absorption spectrum. So in particular, we now must take into account not only the dynamics on the final electronic state, but also on the initial state, since it isn't stationary anymore. And thus we get our transient X-ray absorption spectrum by the Fourier transformation of the dipole-dipole correlation function, where now both the bar and the ket are time dependent. Yeah, to make life a little bit easier, the short-time approximation we want to discuss states that due to the very short-living nature of core excited states, the wave packet evolution is insignificant during the transition. So mathematically spoken, it states that the transition dipole operator and propagator commute. And if we substitute this condition into our expression for the full dynamical dipole-dipole correlation function, we end up with a correlation function where the kinetic energy operator is cancelled out. So in other words, the short-time approximation completely neglects any dynamics after X-ray interrogation, and we can find this equivalent expression for the energy domain. And since so far most theoretical studies of time-resolved X-ray absorption spectroscopy are done within the Lorentzian limit, we wanted to know how good this approximation really works, or rather what we are actually missing when applying it. And for this purpose, we set up a Hamiltonian containing all relevant states and performed two sets of calculations. One using the full dynamical approach, and one where we deleted the kinetic energy operator after X-ray interaction, while all the rest remains the same. So in particular, with this, we've got a clear comparison of the two approaches without having any further approximations. And for this, we needed a test system, which was pyrazine. And yeah, pyrazine is a typical benchmark system for such kind of theoretical studies, since it has an interesting UV photochemical behavior with several conical intersections between the valence excited states, but it is also a relatively small, highly symmetrical molecule, which makes it easier to handle. And yeah, its UV induced dynamics is already well described by several theoretical models. So we actually know what we in principle have to expect and can hence validate our model for the study. And in our calculations, we assumed a core hole lifetime of 8 femtoseconds, which is consistent with experimental measurements at the KEH of molecular nitrogen. Yeah, as mentioned, we want to calculate our spectra within a full time domain approach using wave packet dynamics. And for the nuclear dynamics simulations, we used the MCTDH method, which is a grid-based full quantum mechanical method counting as the golden standard under quantum dynamics simulations. And this method requires pre-calculated electronic states. So we firstly set up a molecular Hamiltonian where we assumed that the valence and core excited state manifolds are completely decoupled, which is justified by the huge energy difference between them. For the valence excited states, we used an already existing validated model and extended this with 3 more core excited states. And for the core excited states, we used the linear vibronic coupling approach to approximate the Hamiltonian, yielding a very good fit, as you can see, for example, here along along one of the symmetric vibrational normal modes. So yeah, okay, let's come to some results. Yeah, before we look at actual spectra, it is worth to consider the valence excited state population, which is strongly connected to the photochemical dynamics we want to track. So here, what we see is that so the yellow S3 state was initially populated through an instantaneous vertical excitation at time 0. And then the population of S3 rapidly decays into both the red S2 state and the green S1 state, which can be explained by low-lying conical intersections between both pairs of states. And after approximately 55 femtoseconds, S3 is almost depopulated, but the population dynamics between S1 and S2 keeps oscillating with a period of approximately 65 femtoseconds. And indeed, this population dynamics is accurately mapped by the transient X-ray absorption spectra, which are shown here for time delays of 0, 20, 35, and 55 femtoseconds. So the contributions of the transitions from each valence excited state are here highlighted by the same color as used in the figure for the state population. So what do we see? Obviously, immediately after the pump excitation, only the excitation band starting from S3 can be seen. And after 20 femtoseconds, the population is almost equally distributed over the 3 states, and contributions of all 3 transitions can be seen in the spectrum. Moreover, for the green S1 state, the population reached its maximum at about 35 femtoseconds, and for the red S2 state at about 55 femtoseconds. And this is exactly what we see in the corresponding spectra on the right-hand side. So we have this band maximum for the green S1 state at 35 femtoseconds and for the red S2 state at 55 femtoseconds. These spectra were calculated including any dynamics. But now we can also see the resulting spectra within the Lorentzian limit shown by the red curve. So the overall shape of the absorption bands within the Lorentzian limits is in good agreement with the full time-dependent calculations. And generally, the excitation bands have a rather broad and asymmetrical shape for both approaches, which must be hence traced back to the evolution of the nuclear wave packets on the valence excited states. However, as we have seen before, the coherent wave packet dynamics after a probe excitation in both the valence and core excited state manifold is only covered by the fully time-dependent approach. Whereas the Lorentzian limit approximation is only sensitive to the probability distribution of the valence states wave packet. And this is why the spectra within the Lorentzian limit lack finer vibronic structure, as we can, for example, see here in both dominant absorption bands for a time delay of 20 femtoseconds. So now I want to come to the second part of my talk and discuss the influence of the external electric field and why we needed delta pulses for a clear comparison between the Lorentzian limit and the full dynamical approach. We now want to go beyond this picture and include an explicit description of the external electric field. For this, we treat the electromagnetic field as Gaussian-shaped laser pulses and use a non-perturbative approach to calculate our spectra. And one can show that the spectrum is proportional to a response function, which in turn is defined by the product of the Fourier transformation of the expectation value of the transition dipole operator and the Fourier transformation of the electric field. Now we can see the effect of a finite pump pulse duration on the X-ray probe response. So along the rows, we increased the pump pulse duration from approximately 1 femtosecond to approximately 80 femtoseconds. And for each row, we show the valence excited state population dynamics, the linear UV absorption spectrum, and transient X-ray absorption spectra for 4 different time delays. And yeah, in the pyrazine system, we have this oscillatory behavior between S1 and S2, as I mentioned earlier. And as you can see, for pulses shorter than 20 femtoseconds, the 2 dominant absorption bands reproduce this very well. So we see this up and down of the 2 dominant bands. However, since the excitation process lasts for the entire pulse duration, longer pulses might lead to blurring effects. And yeah, for pump pulses longer than 50 femtoseconds, it becomes nearly impossible to extract relevant information about the underlying dynamics, and the transient absorption spectra seem to be nearly time independent, as we can see in the last row here. Yeah, so far I restricted my analysis to excited state transitions only, but of course in real pump probe experiments, we can't neglect contributions from the ground state. So this is why I finally show the calculated differential absorption spectra as we would get them from experiments, where the positive contributions stem from the excited state absorption and the negative signals are due to the ground state bleach. Yeah, for pyrazine, we see that the X-ray transition from the ground state is much more likely than from the excited states, leading to a very dominant negative bleach signal. However, this dominant bleach signal doesn't overlap with the time-dependent signals, and we can thus still see this typical oscillating behavior of the 2 dominant excited state absorption bands. So to summarize my main part, we have seen the power of the full time domain approach, as it can avoid the short time approximation without having more effort, as well as include an explicit description of the external electromagnetic field, which is needed to accurately reproduce the situation in the lab. While we have seen that the Lorentzian limit works okay for time-resolved absorption spectroscopy, it might cause larger errors when simulating Riggs experiments, as here the core excited states are immediate states and the valence excited states are the final one. Sorry. So yeah, so an ongoing project is actually to extend the full time domain approach to Riggs simulations. And yeah, as there are a few minutes left, I want to now give a brief overview of how to simulate Riggs experiments using wave packet dynamics. And I will also shortly discuss the advantages of a full time-dependent approach. So in general, resonant inelastic X-ray scattering is a coherent scattering process where the system is firstly excited by a X-ray photon onto the short-lived core-excited state manifold, and then it continuously decays into the valence excited states as well as back to the ground state. Hence, we will later see that our Riggs cross-section is dependent on 2 frequencies, namely the incoming photon frequency, omega, and the scattering frequency, omega prime. And yeah, Riggs complements absorption spectroscopy methods, as for example, one might reach valence excited states which are not directly accessible in the UV range. And also in Riggs, the spectral resolution is not limited by the large core whole lifetime broadening as it is in X-ray absorption spectroscopy. And hence Riggs can also deliver information on the nuclear motion through vibrational substructure of electronic transitions. And the conventional approach for calculating Riggs experiments is cast in the energy rather than the time domain. So generally the Riggs cross-section is, as already mentioned, dependent on both the incoming and scattering photon, and it is proportional to the squared polarizability. In the energy domain, the polarizability is given by Kramer's Heisenberg formula, where 0 denotes the ground state, I is the intermediate core-excited states, and F is the final ground or valence excited states. So as before, for absorption spectroscopy, we can derive an equivalent time-dependent expression. And in the time domain, the polarizability is given by the Fourier transformation of the autocorrelation function of the so-called Raman wave function. So the Raman wave function is independent of time and can be understood as the state from which the emission takes place. That means if we compare the 2-photon Riggs process with the 1-photon absorption process, the Raman wave packet just replaced the initial wave packet when calculating the spectra. In Riggs experiments, the effective lifetime on the core-excited state manifold can be controlled by detuning the excitation from resonance. So in a bit more detail, there's a reciprocal relationship between the time interval that contributes to the Raman wave function and the detuning delta omega. And hence detuning may serve as kind of a molecular clock as it controls the time interval of the dynamics. And these effects can be clearly explained by the nature of the Raman wave function, which also means that the Raman wave function is not only a mathematical construct, but it has indeed interesting physical interpretations. So there's one last thing I want to point out before I finish my talk, namely both Kramer's Heisenberg formula and the Raman wave function approach are only valid if you consider a continuous wave experiment. So if you really have a monochromatic wave. But of course, that's not the case in real experiments. And but by using a full time-dependent approach, we can easily include a proper description of the external electric field, as I have already shown for transient X-ray absorption spectroscopy. And by including this proper description of the field, we can then study effects that stem from a finite pulse duration, and we may also figure out how long a pulse must be to count as a continuous wave, which in turn might be important for future experiments. Furthermore, there's a substantial interest to apply Riggs in pan-probe spectroscopy. And with a full time domain approach, we should be able to extend our method to time-resolved Riggs, like we did for XAS, by including any dynamics on the core as well as on the valence excited state manifold. And last but not least, using wave packet dynamics with an explicit description of the external field will also allow us to explore stimulated X-ray emission processes as accurate as possible. And I hope there will be some results on pyrazine very soon. But for now, I want to leave it as an outlook. And before I eventually finish my talk, I want to thank both groups in Hamburg and Heidelberg. And special thanks goes to David and my supervisors Niels and Oriol. And of course, thank you for listening.
So thank you so much for your talk. Are there questions, comments? Do feel free to write in the Q&A tab or raise your hand if you wish. And we're looking forward to your talk, to your questions. People are quiet sometimes. No? Yeah, people are quiet. But I mean, you mentioned that there is a difference between Riggs and the standard spectroscopy that you were discussing before, and you said that there were transitions that were not accessible that you would like to have a look at. Could you be more specific?
Yeah, of course. So for example, in pyrazine, here some surfaces. So the two low-lying core-excited states are almost degenerate. So I mean, in this figure, we just see one, but there are actually two, and we see non-adiabatic transitions even in this very short core hole lifetime, and yeah, this allows different transitions. I mean, if we just have a transition to one state, it will go back to valence excited state, which would be or probably also reachable by directly from the ground state, but by having this non-adiabatic transitions to a state of different symmetry, we will also get transitions which are allowed by the symmetry rules, which would be forbidden directly from the ground state. So this is one example where how we could reach valence excited states that are not directly accessible from the ground state.
Okay, and this allows then tracing the dynamics.
Mm-hmm.
Very good. Okay, thank you so much. Are there questions, comments? Come on. I'm grilling everybody here. Nobody asks anything. No? Okay. But it was a great talk. Great to hear you. And we're going to move then to our last speaker, who is Dr. Xinyao Liu from ICFO. So we had plenty of people from Germany today. And today, now to close, we have someone from Barcelona. He is a postdoc at the group of Jens Bigert. And he is going to talk about atomic image of complex and chiral molecular structure with laser-induced electron diffraction and machine learning. So one more machine learning talk. He has published this in a quite fancy journal in one of the Nature group. And the difficulty that they were facing is to extract molecular configurations from measured diffraction patterns. And they have established a machine learning based laser-induced electron diffraction framework to overcome these issues and to retrieve the atomic position of 1D, 2D, and complex 3D chiral molecules. So it's quite a hot topic, in fact, and we're looking forward to your talk. He's going to talk about a molecular selfie, which is LEED, laser-induced electron diffraction. And we're also thankful for your picture. You sent a picture of you hiking, so we asked people to send fun facts. So we learned that you quite enjoy hiking. A really nice picture. So we're ready to listen to what you have to say. We're ready to go. The floor is yours. Feel free to start whenever you like.
Thanks for the nice introduction. My name is Xinyao. I come from the ICFO Institute of Photonic Science in Barcelona, led by the group leader Jens Bigert. I will talk about the atomic image of the complex molecular structure with laser-induced electron diffraction. Okay. So why we want to image the molecular dynamics? One of the most excited biological process is the formation of the vision. Retina is the key molecule for the formation of the vision. Light can convert it 11-cis retina to the all-trans retina. This is one of the key chemical reaction to let people see the world. DNA, which carry the genetic instructions. People first see the DNA double helix structures from 1953 by the X-ray diffractions. So in order to see the molecular dynamics, we need to have a proper spatial resolutions. Like normal reaction, a microscope can only have the 200 nanometer spatial resolutions. And then for this kind of molecular internuclear distance, this typically in the angstrom level. This is like 10 power of minus 10. And then in the meanwhile, if we want to visualize the molecular dynamic, we also need the proper temporal resolutions. Like for example, the molecular rotation is in the picosecond level, and then the vibration is in the femtosecond level, and then the electrodynamic is in the attosecond level. That means to directly image the ultra-fast dynamic in the isolated molecule, we need angstrom levels spatial resolution and femtosecond temporal resolutions. So typically one of the candidates to image the molecular dynamic is use the photons. For example, the X-ray. X-ray is a high energy electromagnetic radiations, and then its typical wavelength is around 10 nanometers to 10 picometers. And then the X-ray pulse interact with the samples, and then people by analyze the diffraction items can retrieve the molecular structures. Another candidate is we use the electron. For example, the ultra-fast electron diffractions, people use high energy electron pulse interact with the target. By analyzing this electron diffraction patterns, people can still retrieve the molecular structures. But however, for the X-rays, people need a careful balance between the X-ray brightness and the damage threshold of the samples. And then for the electron have much higher cross sections, but due to the space charge issues, sometimes the electron pulse duration is hard to shorter than the 100 femtosecond levels. So any one of the technique can take advantage of the both. So here you are. We have the atomic scale molecular image. We image the single molecule with only one electron. They can reach the picometer spatial resolution and atto to femtosecond temporal resolution, which is called laser-induced electron diffraction. Normally we call this LID. Let's briefly talk about these physical pictures. When the laser interacts with the target, the electron ionized from the target molecule be accelerated, returned, and rescattered with the target molecule. By analyzing this rescattered electron, we can retrieve the molecular structures. We can understand this is a single electron wave package interact with the target molecule. By analyzing the interference patterns in the far field, we can retrieve the internuclear distance. This is a molecular scale electron microscope. But in our case, the electron is not from the external. It's ionized from the target molecule and driven by the laser. So we have the several advantages. For example, the sub-cycle timing is can reach to the attosecond resolution. We don't have the space charge issue because we only have only one electron, and then the cross section of the electron is 5 orders higher than the X-ray. So the whole process is we can understand is also electron is as a single electron diffraction, but the whole progress is driven by the laser. So let's see the experimental setup. The target molecule released by the supersonic gas jet intersected with the mid-air laser pulse, ionized the electron and then the ion fragment. By applying the external electric and the magnetic field, the ion fragment and the electron will be pushed to their own detectors. So we can capture the 3D momentum of the electron and ion simultaneously. It's combined with mid-IR OPC pair laser cells. The wavelength about 3.2 micron, 5 optical cycles, pulse duration around 15 femtoseconds, and then the intensity can higher than 10 power of 14 watt per centimeter squares. And then it's combined with a reaction microscope. It can catch the 3D momentum of the electron and ions coincidentally. That means we know which ion or which electron corresponding to which ion fragment. So this picture is showing the real setup in the lab. The whole system is running on the 10 power of minus 11 millibar without gas load. So LID has successfully retrieved the molecular information from small gas phase of a molecule. For example, the oxygen. Here we use the Fourier transform method, and then by analyze this Fourier transform spectrum, we know by identify the peak position, we know the internuclear distance. And then we also retrieve the acetylene like C2H2. Here we use the fitting routine by finding the best fit between the experiment and the simulation result. we can retrieve molecular structures. And then also the carbon density line. Here is still same as based on the Faking routine. And then furthermore, we also retrieved one of the most simple liquid molecule water. Here we trace the water molecule variation under the different electric field, with the increased the field stress. You can see the OH bond length is increased, and also the HH bond length, and then the bond angle is reduced. Like we resolve the symmetric stress of the field stressed water molecules. Okay, now we already showed the lead have an advanced technique to retrieve the small molecule, but then the further step, like we want to retrieve the more complex and the larger molecule. But now the current retrieval algorithm is limited by only a few atom systems. But in this talk, I want to show like we are able to retrieve the complex molecule structure use machine learning. So the figure is showed the diffraction patterns vary with the different molecular structures. You can see the different molecular structures is corresponding to the different patterns, but the difficulty is how to identify the molecular structure from the diffraction patterns. This means that we need to be able to identify the global extreme from this multi-dimensional space. For the small molecule, it's always easy. We can use like simple this convergence strategy, like we showed it, like use a chi-square fit or multi-peak fit. However, for this complex molecule, we will face the unfavorable scaling issues. And then this is also a challenging for the any diffraction method, like X-ray diffraction and ultra-fast electron diffractions. So here we want to use the machine learning that can simultaneously consideration of the manifold of the degree of the manifold degree of the freedom, and then we use machine learning to try to retrieve these complex molecular structures. And then let me briefly talk about the machine learning. So in the typical neural network, it's combined with 3 layers. Like normally you have the input layer, we feed it into the input value in the first layer, and then in the middle you have the hidden layers, and hidden layers perform the mathematical calculations like the weight assumption, the bias addition, and active functions. And then in the output layer, you generate the output values. And then let's zoom in the one of the neurons in the hidden layers. So here for the neuron in the hidden layers, and then the input value from the first layers, and then the connection between each neurons, we can understand it's a weight functions, because not all of the input values will share the same weight, because always one parameters may be more important than the others. So this weight function is to identify like which parameters is more important. So give the different importance of the different input values. But the calculation is very simple. You just use the omega is the weight parameters multiply the input values and then plus the offset. It's also called bias. And then later on you apply a nonlinear function. Like here we use the omega z. So the one connection between the first layer to the second layer is through the first through the linear calculation, and then you added the nonlinear functions. And then this from the first layer, for example, the first layer to the second layer's neurons. And then you repeat the same calculations until the third layer and then the final layers. So the whole process is quite simple. It's like you through the linear function and then a nonlinear functions. You calculate through the layer by layers until the output layers, and you got output values. So here, what we did, we try to build a machine learning architecture to retrieve our data. The first step, we need to do the whole system is based on the supervised learnings. We first make a database with 100,000 of the molecular structures, and then we simulated their differential cross-section. Here we call it DCS. We can also understand it's kind of a diffraction patterns. And then we split it into the 3 subset, like training side, validation side, and test set to validate machine learning models. Here, like we try to like machine to learn the relationship between the molecular structures and their corresponding the diffraction patterns. Like once the machine learning can understand the relationship and he know like which diffraction patterns corresponding to the which molecular structures, and then we put the experimental diffraction patterns to the machine learning models, then the machine learning models will give us the predict molecular structures from the experiment. The step is first we pre-calculate sufficiently large ensembles to validate machine learning models. The database we run is through our internal clusters with 1,000 parallel calculations. This normally take around a couple of weeks. And then the training process we running on the Google Cloud with 100 GPU. This normally take a couple of hours. And then the machine learning, for the machine learning models, we specifically use like convolutional neural network. This is well suited for the problem of the image recognition or facial recognition. This is also one of the foundation algorithm to unlock your mobile phone using the Face ID. So let's go detail about how the convolutional neural network and then it's work for our molecular retrieval. So we have a pair of the input, the molecular structure, and their corresponding differential patterns. So we first feed the differential patterns to the machine learning models. The differential patterns will first convolute with different filters to generate the feature maps. The 2D differential patterns will the source pixels will convolute with the convolution filters and then generate the destination pixels. And then the convolution filters will convolute the whole pictures of the input map and then generate the feature maps. And then they will use different filters to repeat the same process until you generate a collection of the feature maps. These feature maps will capture the subtle feature of the input pattern. It will then we use different filters to capture the subtle feature of your input patterns. And then we feed it into the traditional fully connected neural network, as we previously talked about it. And then this through the weight and then bias, this will give us the predict molecular structures. And then the machine learning is an iteration algorithm. In the beginning, all of the parameters are initialized, randomly initialized. And then after each iteration, they will simultaneously optimize all of the parameters. And then they try to minimize the difference between the predict value and then the real value. So here we use the cost function to evaluate the difference between the predict value and then the real values. Here we just use an example with only two parameters. We can see in the beginning, the cost function, that is the predict value, it's really far away from the real value because it's randomly initialized in the beginning. And then after 5 iterations, we see the cost function is become smaller, smaller, smaller. Then this means the predict value is almost the same as the real value. Then we can conclude that the machine learning are able to predict the value is same as your input. Then the machine learning are able to get to the relationship between the molecular structure and then the differential patterns. So this generates the two-step. The first, we use the 2D differential cross-section to convolute with different filters and then multiply the weight between each neuron to get the predict atomic position. And then by the iteration loop, the machine learning model can able to predict atomic position. It's the same as the real values. And then we can go to some detail about the training process. Here we use the mean absolute error to evaluate the difference between the predict and the real values. We can see after 15th iteration, the three database, the three training side, like training side, validation side, and the test side will go to the minimum. That means the machine learning predict value is the same as the real values. Here we also use the correlation map. It's correlated with experimental patterns. with the simulation pattern from the predicted molecular structures. So we can see it's stronger correlated. And then after that, we try to extract molecular structures. We use the machine learning models. So we start from the beginning with the easy and simple 1D molecule, C2H2 carbon acetylene. So we first make a database, like we first simulate all of the possible molecular structures and calculate through our clusters. For example, here we can change the bond length of the CC, and then we have also changed the bond length of the CH. And then we calculate 100,000 of the possible molecular structures, and then we also calculate their corresponding the differential patterns. And then we feed it into the machine learning models and calculate it through the Google Cloud. Then the machine learning models try to identify the relation between the molecular structures and then their differential patterns. And then after the 15 times iteration, the machine learning model can understand the relation. And then after that, we put our experimental differential pattern to the machine learning models. Then the model will give us the predict molecular structures. And then we compare with our previous publication. It's based on the fitting routine. We can see they reached the excellent agreement. And then we go to one further step. We go to a 2D molecule as carbon dioxide. So here it's the same, but it's a little bit complex. It's 2D molecule. Here we vary not only the bond length, but also the bond angle. The same, we calculate all of the possibility of the molecular structures, and then we calculate their differential patterns. We feed it into the machine learning model, try to make that machine learning model to learn, and then get the predict value is same as the value we input. So we training the machine learning model on the simulation database. After that, after the machine learning model is validated, we put the experimental differential pattern to the machine learning model. Then this gives us the predict molecular structures. So we can see it's the we also compare with the previous publication based on the fitting routine. You can see the bond length is almost the same and the angle is almost the same. So they also reached the great agreement. And then we want to do one step further. We want to retrieve a more complex molecule. So here we choose a candidate. It's called benzyl. It's 27 atoms. So if we use the traditional method to retrieve the molecular structures, we will face a big challenge. For example, previously we used the Fourier transform based on the Fourier transform method. Here we need to identify the peak position, because the peak position is indicate the internuclear distance. For example, this molecule has a 27 atom. That means you have the more than 100 internuclear distance. And then if you want to see the Fourier spectrum, you will see all of the bond lengths will be overlapped. And then maybe some bond lengths is isolated, but you have the multiple solutions. You don't know like which is indicated to the which internuclear distance. And then another method we typically use is the fit based on the fitting routine. However, this is another challenging because like the small molecule, we can calculate all of the possible molecular configurations. But for this complex molecule, we cannot calculate all of the possible molecular configurations. Then this will require a huge calculation time. It's not really realistic. So this two methods like we typically use will not work for the complex molecule. So here we want to do something different. What we did, we divided the molecule to the 4 subgroups. And then we considered the variation of the 4 groups, and then we also considered the global change of the molecule. For example, we considered all of the bond lengths is increased 10%, 20%, and 50%. And also all of the bond lengths is shrinking like 10%, 20%, and 50%. For example, in this 4 group, for example, in the group 1, we only consider the bond lengths is stretching and decrease. And then for the group 2 is the methyl group, we consider the variation of the bond angle and then the bond length. For example, this group 3, we only consider the angle change. And for the group 4, we consider the bond length change and then the bond angle change. So we try to train the machine learning model on this interpreted sufficient reduced database. We try to avoid calculating all of the possibilities because it's not realistic. It requires huge calculation time. So we train the machine learning model on this reduced database. We try to ask the machine learning models to understand the relationship between the molecular structure and their corresponding differential patterns. And let's see. So here we can see after 100 times training, we see the mean absolute error. This is the error between the predict value and then the real values. You can see after 100 times training, it's reached to the minimum. That means the machine learning model can able to predict value is same as your input. And then after we finalize the machine learning model training based on the simulation database, and then we put on the experimental 2D DCS, the differential patterns to the machine learning models. This gave us a predict molecular structures. So here we predict 7 atom position with X, Y, Z coordinated. Because we predict the 3D position of the 7 atom position. And then there's slightly difference between the predict value and the equilibrium value is due to the laser field induced. With this predict 7 atomic structure position, we can reconstruct the molecular structure of the function. And then the green area is presented by the error bar. So we can see we successfully retrieve the molecular structures based on the machine learning. Because compared with other traditional techniques, machine learning is able to train on this sufficient reduced interpreted database, not calculate all of the possibility. And we focus only 4 groups of the atoms, and we also consider the molecular-wide global change of the structures depending on this interpreted database, and then reconstruct the molecular structures. So machine learning combined with LEAP overcome this scaling limitation in this standard fitting routine, and it's achieved high accuracy pattern matchings in the complex solution space. We successfully retrieved position of the atom like 1D acetylene, 2D carbon dioxide, and complex 3D molecule functional. We need to also mention this the limitation is not only for the LEAP diffraction patterns, but for any diffraction method. It provides our new opportunity to image complex and larger molecules in the static and time resolved studies. Here I want to specifically thank my professor Freibert with his support, and also my teams. And also I would like to thank the whole group in the ICFO and in the atomic, like ultra-fast optic groups. Okay, thank you.
So thank you so much for this really nice talk. I can see we have a few questions. The first one is from an anonymous attendee. Could you expand on how does the model simulate the lead process from the position of the atoms to then compare results with the experimental ones?
I don't really got the answer.
Yeah, can you elaborate on that? I know you're anonymous, but maybe you could type a little bit more about what you mean. No? Yeah, no, I don't know.
Maybe I try my understanding.
Yeah, give it a try, your understanding, and then you can, the anonymous person can say if this is fine or not.
Yes.
I think you want to know how you simulate the process, because you had QRS and then you had the machine learning bits, right?
Yes.
You mentioned that list.
Yeah, so here we first calculate the simulation database. This is based on the IAM independent atomic models. So we first simulated we simulated the diffraction patterns, use the independent atomic models. And then, because in the lead process, we assume the returning electron has super high energy, and then everything can be used based on the independent atomic model. So we use the independent atomic model to simulate the diffraction patterns. And then we change the different molecular structures, like we think it's all possible molecular structures, and then we simulated like 100,000 molecular structures and their corresponding diffraction patterns. And then we use this as a database to training on the machine learning. So the lead process, I think, is based because we use high energy, because we have the longer wavelengths, and then the returning electron have super high energy. So everything can be based on the independent atomic model.
Thank you. I think he's fine. Now, Manfred Lai, could you explain what is the independent atomic model? Thank you.
Okay, so because independent atomic model, like it's ignore the correlation between atoms and atoms in the molecule. For example, in the molecule, you have several atoms, but using this, for example, you have 3 atoms, like the water, and then you assume these 3 atoms is independent. There's no correlation between the hydrogen and oxygen. The 3 atoms is independent, but this only assumption work when you have the high input probe beam, like we use higher energy is higher than, normally we say higher than 50 eV, and then we can treat it, the atom is independent. There's no correlation between, or stronger correlation between each other.
Is that clear? Manfred, if you would like to ask further, I have allowed you to talk, so you can pursue further and you can mute yourself.
Can you hear me?
Yes.
Very good. Okay. Well, I thank you for the answer and thank you for the nice talk. I understand what you said about the independent model, but I mean, my question is just what are the equations that are solved? I mean, you treat the atoms independent, but I mean, I don't know what kind of equation is solved. So this, I understand, it does not include a strong laser field, I suppose. I suppose it's some kind of scattering calculation. And if that is the case, I'm wondering how this is extracted from the strong field electron distribution, right? Because, you know, I mean, you have to do something to extract the scattering cross-section from those experimental data, right?
Yes. So you mean the equation, for example, between the diffraction pattern and then the internuclear distance?
Yeah, I mean, so how do you simulate the DCS, right? That's my question.
So normally we simulated like, it's like make the sum of the scattering amplitude F, multiply another scattering amplitude of another atom, and then
okay. And where does that come from? This is clear, but where does the F come from? I mean, I don't. It doesn't come from the textbooks, right?
You mean the scattering amplitude?
Yes.
I think it's based on you have the planar wave electron wave package interact with the single atoms.
Okay, so you do this with your own code, or you use some package to generate results?
We have the code. It's called LSEPA.
Okay.
And then we use this code to do the simulation. Okay.
And then my other question would be how this is related to the photoelectron distributions, because, you know, if you have a scattering picture, this will be somehow shifted by the strong field. So, I mean, I think you have to do something to relate that to each other.
Yeah, because when we retrieval the DCS, we call it field-free DCS. Because during the retrieval process, we remove the shifted from the laser field. Like our scatter momentum is equal to the field shifted, like vector potential shift from the laser, and then the return momentum. And then when we do the data analyze, we subtract the shifted from the laser, and then we call it field-free differential cross-section.
Mhm. Okay, thank you. I've asked enough. Thank you very much.
Thank you.
We have another one from Anonymous. What is your training set based on? Theoretical predictions or previously collected experimental data?
So the training database is only the simulation. So the training base is like we simulated 100,000 of the molecular structures. Like we first like use a Python code to write it, all of the possible molecular structures. For example, your bond length can increase 100%, but it's not cannot like increase to 1,000 like 1,000%. Like for example, we can double the bond length, but you cannot say like you cannot say triple or quadruple the bond length. Sometimes we put in the realistic range. For example, the maximum we say like 20%, 200% bond length. For example, we varied all of the possible bond lengths, and then from there we use the independent atomic model to simulate the diffraction patterns. Like we first to like make a database of all of the possible molecular structures, and then like 100,000 molecular structures, and then we used our cluster to calculate all of the possible diffraction patterns, and then we use this as a database.
Is that clear? Yes? Seems fine. Okay, thank you. I mean, I think there are no more questions.
I would actually have one question.
Simon, I would also have a question. Okay, unmute yourself. Go ahead.
So in the same direction like Manfred, so if you use your photoelectron momentum distribution, what are you exactly looking at? So I understand that you try to reconstruct the field-free DCS, but in general, over the scattering plateau, you have the interference, for example, between long and short rescattering trajectory. So you cannot easily disentangle the DCS or both contributions. So in principle, I would expect that you can only use the outer part where long and short trajectory merges. And hence, when you do it, you basically get a one-dimensional observable, namely a DCS is a function of one angle. And then how can you use a one-dimensional observable to reconstruct such high-dimensional things like atomic positions where you have 5 atoms or even more? So how can this be related to each other, and how can you be sure that you get out the correct result and not something, yeah, how can you make sure that it is a unique result?
So here you mentioned the long and the short trajectory for this, yes. So, but the long and the short trajectory is a quite long story. But in here we assume like most of our rescattered electrons from the long trajectory due to the ionization rate. But you know the long trajectory, the release time is more close to the peak of the field. So we assume like most of our electrons come from the long trajectory. And then when we selected the differential cross section, the DCS, we normally selected from 2UP to 10UP, and then we selected like the 2D. Then we can make sure like we kind of get rid of the photoelectrons, and then we selected the region of the from 2UP to 10UP as the rescattered electron regions. And then we assume it's based on the long trajectory due to the ionization rate.
Okay, but then when you try to extract something, you have to get rid of the ionization rate, for example. And I would expect that for such a structured molecule, it is not trivial to know the ionization rate over the scattering plateau. So
yes, but when we do the measurement, we also can monitor the count rate. For example, the ionization rate is also related to the count rate. For example, we have the 106 kilohertz pulse, 106 kilohertz laser system, and then we only measure the electron count is roughly 20K or 30K. Then it's kind of 10. You have the 10 pulse, and then you only have 2 ionization happen. So we try to not ionization. Like we try to like do a single pulse ionization. Like you have the 10 pulse, and then you only have 2 events to analyze. Like we assume it's always in the single ionization, single charge ionization, and then the intensity is not that high. So like try to minimize this effect.
Yeah, what I mean was more that for different momenta over the scattering plateau, you could have a different ionization rate, for example.
Yes, yes.
But it is fine. It is okay. So thank you.
Thank you.
Thank you everyone for joining. If you would like to ask questions more privately, just hang in there. We're going to stop our transmission soon. So for those who are watching on YouTube, and there were quite a few of you today, take good care of yourselves. Have a great Christmas, New Year, holidays, relax, and we will be back in late January. So take good care of yourselves. If you would like to ask questions, but you're a bit shy, just hang in there because you have the chance, and see you soon. Thank you for joining and supporting our early career researchers.